So I'm going to talk to you guys today about some work that I did in close collaboration with two students. So I want to acknowledge them right from the start. My PhD student, Amy Patterson, and a recent graduate, Alex Strang, collaborated on all of the work I'm going to show you. So the question that we were interested in was understanding when and where we can expect to see early warning signals in multi-species systems approaching tipping points. And of course, those of you who are in the pedagogical school have just spent a whole week thinking about tipping points. Everyone else is going to spend this week thinking about tipping points. So from an ecological standpoint, what we're talking about with tipping points is the idea of a sudden change in the ecosystem state in something qualitative and observable. So something that ecologists would sort of care about. And so some classic examples are shown here in the photos. Coral reefs can be either coral dominated or algal dominated. These photos show sort of a classic grassland woodland transition in terrestrial ecosystems. And here we have two adjacent lakes, actually with an artificial barrier between them, because these are experimental lakes. So we're seeing on this side, again, algal dominated versus a clear water state. So these are all examples of different sides of a tipping point. And the type of tipping I'm going to be talking about today is B-tipping. So these are tipping points associated with a bifurcation that alters the stability of the current state and then prompts a tip into the alternative state. So critical slowing down, of course, occurs before certain tipping points. And so we're looking at here is a bifurcation diagram with respect to some bifurcation parameter. There are two tipping points visible in this diagram. And if we think about what the eigenvalues are doing in approach to the tipping point, where of course the dominant eigenvalue is giving the negative return rate. So a negative value is returned to equilibrium slower if it's closer to zero, or faster if it's more negative, and a positive value is the speed of divergence again slower if it's closer to zero. And the eigenvalues associated with this diagram I'm showing up top look like this. So we, of course, cross zero at the two tipping points. And these arrows going from a greener color to a redder color in both diagrams represent the speed of return to the equilibrium. And so where it's greener and this dominant eigenvalue is more negative, we get a faster return, which we can think of as a ball rolling faster on this diagram down to a stable well. The orange or colors are an intermediate rate of return, and then right before the bifurcation, this well is quite flat and the rate of return will be quite slow. So this is critical slowing down. It occurs following a perturbation near a tipping point. So importantly here, we need to be perturbed away from the equilibrium value in order to have the opportunity to observe critical slowing down. So again, those of you who were in last week's school, I believe Partha gave you a really nice introduction to early warning signals. So these are statistical symptoms of the critical slowing down. If we gradually change a bifurcation parameter through time, and so here we're plotting two things. The blue is the bifurcation diagram, and the black is a simulated trajectory of the population dynamics through time. In approach to a tipping point, where again, farther away from the tipping point, these stable equilibria can be thought of as having these deeper, more stable wells, faster return, and closer to the tipping point, this higher equilibrium is still stable, but just barely. There's a well here, but it's almost flat, slower return. So when we have this sort of flatter part of the landscape, we can have longer forays away from equilibrium, and the result of that will to increase the temporal variance and increase the autocorrelation, because there's a longer duration of return, sort of in a net one directional flow back towards the equilibrium. There are other statistical warning symptoms of critical slowing down that count as early warning signals. I believe you guys discussed several last week. These are the two that I'm going to emphasize in my talk today. So this is just an example zooming in on this portion of the time series of this black simulation. We see a rise in autocorrelation and a rise in variance in advance of hitting this tipping point. So there's an important role of noise or stochasticity in this process. and an important role of determinism. Of course, the tipping point itself, the bifurcation, is related to the underlying deterministic structure of the model or of the ecosystem that it represents. But the noise is what allows us to see early warning signals. So of course, if there's enough noise, we can get variance that's just related to noise and not related to critical slowing down, and that could mask an early warning signal. But noise is also essential. It doesn't just mask early warning signals. It's actually essential to allow us to see these early warning signals, because without it, we would just track the equilibrium. We only see the signals as the system is responding to being perturbed away from equilibrium. So no perturbations, no early warning signals. So when we're talking about early warning signals, we're really talking about a nice example of the interplay between stochastic and deterministic aspects of ecological dynamics. So these early warning signals have been demonstrated in real systems. There's a nice demonstration by Dye et al. In 2012 of critical slowing down and early warning signals in a lab system. These authors grew this budding yeast in very small populations in these 96 well plates. So lots of replicated populations of budding yeast under different dilution factors. So they're diluting the media that these yeast are growing in, which acts as a mortality. Higher dilution means more mortality because you're taking out some of the yeast and putting in just more solution without the cells in it. This is an empirically based bifurcation diagram showing a stable fixed point and an unstable fixed point based on their experimental results. And they show a clear rise they show it as the standard deviation, but of course, this is also a rise in variance in the temporal dynamics of the cell counts as they approach the tipping point along this stable line. So just to demonstrate to you guys that this is not just a mathematical phenomenon, it's something that we can observe in real systems as well. Okay, so everything I've talked about so far, we can sort of imagine it applying to a single species, a single population. But the premise of the work I'm presenting here was wondering about how critical slowing down appears in systems with multiple species. So if we're just talking about one species, to orient you to the more familiar way of thinking about this, if we just had a single species and we're looking at the probability distribution or frequency distribution of observations of that species, in the deterministic sort of hypothetical, no noise, we would have, presuming we've given it time to reach equilibrium, we would just observe the equilibrium. Of course, with some noise, suppose we're now far from the tipping point, we see some distribution of population sizes around this no noise expectation. And then closer to the tipping point, the rise in variance means this distribution is going to spread out. Now we can think of this, of course, in higher dimension. I'm not going to draw more than 2D because I don't really know how to do that, but this is a two-dimensional, you can think of this shape as being a hump in 2D space, or your imagination, your ability to picture this in higher dimension is the only limit. Of course, the same principles would apply. So to make this kind of distribution easier to look at, I'm going to show you some graphs that look like this. When representing the deterministic equilibrium with the black dot, and then the darker shades are the denser parts, or closer to the peak of the probability distribution. So in a two-species system, we could think about the distribution of population sizes with noise as being represented by something like what I've drawn here. But of course, critical slowing down is a directional phenomenon. I drew on the last slide a nice symmetrical set of circles, but in fact, in real systems, as the variance increases towards a tipping point, it's not going to increase equally in all directions. And specifically, it's going to increase the most along what I refer to in this work as the CSD, or critical slowing down direction. And that corresponds to the dominant right eigenvector of the linear dynamics that describe what's going on at this equilibrium point. So critical slowing down is one of the things you'll see with critical slowing down is an increase in variance, but specifically the variance is increasing in this direction in the phase space. And then if that's kind of what it would look like for one particular set of parameters, but if we have some sort of bifurcation parameter that's changing, maybe out here is where the equilibrium and the CSD direction and the distribution of population sizes given some noise, maybe it looks like this away from the tipping point, and as we approach the tipping point, we see that the variance gets more and more spread out in this CSD direction. So that means that because critical slowing down is a directional phenomenon, our ability to observe it is also directional. So to maximize early warning signal detection, we would want data along this axis, along this CSD direction. Ideally, but of course that's not realistic. This is some linear combination of population sizes, and that's not a directly observable thing. So what's more realistic and what is more typically done in ecology is that we observe one or several species. So here I'm drawing the observation direction if we were just monitoring, say, species 1. And in that case, we are not observing directly along the CST direction, but we're observing along a vector whose relationship to the CST direction we could describe. So as it turns out, the angle between the CSD direction and the observation direction will in fact determine the strength of early warning signals and therefore our ability to, the strength of early warning signals that are visible in the data, given, data along this observation direction. And specifically, the smaller this angle, the closer these vectors are to one another, the stronger the early warning signals for any critical slowing down that's occurring will be. So I'm going to show you an example. The work that I'm presenting here was very much inspired by a nice paper from Boylist and colleagues in 2013 in PLOS One. They presented a model of two fish species, predator and prey. So there's a predator population, and then the prey population is split into an adult class and a juvenile class. And the predators only eat the adults. So the way the model works, this is their model. There is a birth term, a maturation term, there's a predation term, and then there's mortality out of each of these classes. I'm going to use as they did, I'm going to follow their lead and use the mortality rate of the predator as my bifurcation parameter. And if we plot that there, of course, this is a three-dimensional model. I'm just showing you one of those state variables here. I'll show you a three-dimensional version of the bifurcation diagram in a moment. But here we have a range of alternative stable states and 2 tipping points. And what I'm going to show you on the next slide is an animation of what all 3 state variables here. It's an animated 3D, so it's in 3D space, a trajectory through time. It will draw out through time as I gradually increase the predator death rate to drive us across this tipping point. Okay, so just so we can see how it works. So that's this animation. We have our 3 state variables, and it's drawing out the trajectory through time. This is critical slowing down. You see how it slowed down right before hitting the tipping point, and switching to the alternative stable state with high juvenile density, which is what we were looking at in the previous slide. So I know that's a little bit of a hard thing to see. I think it's kind of an instructive animation, but let's dig into what the individual, so we don't have to look at 3D space, what the individual state variables were doing through time. The juvenile density was increasing, the adult density were increasing. These are the 2 prey categories, and then the predator density is decreasing as we move our bifurcation parameter toward the tipping point. So in the piece of the time series I'm showing you here, I've cut it off before we actually cross the tipping point. And we can see that in the juvenile population, there's a clear increase in variance here. And that is because the juvenile state variable is close to the critical slowing down direction. So there's a small angle between this observation direction and the CSD direction. But for the other variables in this system, they are much different than the CSD direction, and we see no increase in variance. So as the authors, Borlist et al., presented in their paper that inspired our work, if you're not monitoring the right variable, in this case, juvenile density, you will not see any early warning signals, even though critical slowing down is occurring. So this really raises the point that there's great importance in monitoring the right species in a multi-species community. So the angle between the observation direction and the critical slowing down direction will determine the strength of early warning signals that you can observe, where stronger early warning signals occur for species whose axes are closer to parallel with the critical slowing down direction. So in a two species example here, we'll just start by looking at species 1. If this is just giving you a baseline, how it returns to equilibrium when it's far from the tipping point, we can compare that to how it returns when it's close to the tipping point. In this case, species 1 has a small angle between, if we're observing species 1 and the CSD direction, and we see a clear instance of critical slowing down, much slower return to equilibrium for this species close to the tipping point. In contrast, exact same system, but now suppose we are observing species 2. So this is our observation direction instead. Again, this is what it looks like far from the tipping point, just as a baseline. Large angle near the tipping point between our observation direction now and this critical slowing down direction. And now we see no evidence that there's critical slowing down here because our observation direction is too far from the direction in which the variance is increasing, the CSD direction. So to try to understand which species we should monitor, we clearly know it matters which species we monitor. We don't have the opportunity to observe early warning signals equally in all species. So what should we watch if we're worried about being able to detect early warning signals? We tackled this in two different ways. The first is Amy's work, and the second is my student Alex's work. I'll present that second. What Amy did was she did a numerical exploration for generic two-species systems. So she imagined two interacting species where their growth rates are governed by some generic nonlinear equations. And we have to remember that critical slowing down is only going to occur the way I've just described it if we're near equilibrium. So we can imagine there's an equilibrium point where these two species coexist. And then we can approximate the dynamics near that equilibrium point with the Jacobian matrix. And I'll call the elements of the Jacobian just A, B, C, D, so they're easy to refer to. What Amy did was she considered 21 to the fourth Jacobians. So she did that by taking all combinations of each A, B, C, and D, ranging from -2 to +2 at increments of 0.02. And she retained those where the Jacobian had negative real dominant eigenvalues. So in other words, we want the cases where there is stable coexistence at a stable point. And for all of those that she retained, she computed the, well, she had the dominant eigenvalue and she computed its right eigenvector, and that gives the CSD direction for each of these Jacobians. Finally, she can compute, again, just using the entries of the Jacobian as the inputs, she can compute this angle between the observation direction, assuming, just for argument's sake, that we're observing species 1. We could, of course, just flip it and observe species 2 and get the same insights. So we're saying, for the sake of argument here, that we're observing species 1, pardon me. We know our CSD direction, and we can calculate the angle between them for all of these Jacobians. So from this exercise, Amy was able to derive some pretty nice rules of thumb. They're a little bit complex, so I'm going to introduce you to them gradually. What I'm going to show you is a series of graphs where we're plotting the value of A, which is this top element of the Jacobian, and the value of D, the bottom, against each other. And in this, below the one-to-one line, of course, where the absolute value of A is less than the absolute value of D, species 2 is the one that is experiencing the stronger intraspecific density dependence. So this is species 1's effect on itself, species 2's effect on itself. So when A is less than D in absolute value, it's species 2, this D, that has the stronger density dependence, and on the other side of the one-to-one line, it's the opposite. And we can consider this in various settings for the remaining parameters, C and B. When B and C have the same sign, we're looking at either a competitive or a mutualistic system. So if both B and C are negative, that's competition between the species. They're each having a negative effect on the other. And when both B and C are positive, that's mutualism. Both are having a beneficial effect on each other. When B and C have opposite signs, we're talking about a consumer resource like a predator-prey type of interaction. One is benefiting from the interaction and one is being harmed by the interaction. And we can look at these in the case where B is greater than C in absolute value. So that's where species 1 is experiencing a stronger effect of its interaction with the other, and vice versa. Okay, so we're ready to see some real results now. I'm going to plot the results using a color scale to represent how big this angle between the observation direction, which is, suppose we're monitoring species 1, and the CST direction. We're going to look at those angles here. When it's bluer, then the CST direction is quite similar, quite close to parallel with the species 1 axis. And when it's oranger, it's quite close to parallel with the species 2 axis. And this is what Amy found. These are all the combinations filled in for a particular set of parameters, except where the white gaps are. That is where there was no negative real eigenvalue. So we can pull out some patterns here. We notice that it's mostly orange above the one-to-one line and mostly blue below. And that tells us that the CST direction is most closely aligning with the species experiencing weaker intraspecific effects. And we also, so in other words, mostly orange here, mostly blue here. We also see that the CST direction aligns most closely with the species experiencing the stronger interspecific effects. And we see that by noticing that the split is not exactly on the one-to-one line, it's shifted. And so when species 1 is experiencing the stronger interspecific effect, it shifts this way. And when species 2 is experiencing the stronger interspecific effect, it shifts the other way. So shifting this way represents species 1 as having a stronger effect from the interaction than species 2 is, and vice versa. So we have 2 rules of thumb here. The first, because the split between orange and blue is closer to the one-to-one line in this competitive or mutualistic case, Amy came up with the recommendation that in competitive and mutualistic systems, this intra-specific rule is sort of the stronger one, the one that has most precedence, the one that gives you the most information. And for antagonistic systems like this consumer resource example down here, it's actually the interspecific effect that is stronger. We're shifting farther away from the one-to-one line. So I know that's a little hard to see in those diagrams, but I did want to show you some real results. We'll summarize what that means in words. Some rules of thumb for maximizing early warning signal detection in a 2-species system are that we should monitor the species experiencing the stronger interspecific effect, so the stronger self-limitation. We should also monitor the species experiencing the weaker intraspecific effect, so weaker competition or weaker mutualism upon itself, etc. Now, if these 2 rules of thumb point us to the same species, that's great. If species 1 has both these things, monitor species 1. If these 2 rules of thumb conflict, so one of us tells us monitor species 1, but the other rule tells us monitor species 2, then we say, if we have a competitive or mutualistic system, probably following this top rule is going to serve you better. And if you have a competitive system, probably following this second rule is going to serve you better. So that summarizes Amy's work. And what I would like to do next is walk you through the extension that my other student, Alex, did. And what Alex noted is that everything I've told you so far assumes isotropic noise. So noise that is equally likely to perturb the system in all different directions. In nature, anisotropy in the noise is likely. And so if we have a primary noise direction, so just for the sake of argument, I've drawn a new vector here that says that species 2 is perturbed a lot more than species one by whatever the source of noise is, then that's going to shift this distribution so that now the axis of greatest variance no longer exactly aligns with the CST direction, because there's variance caused by critical slowing down and variance caused by a primary noise direction, and they sort of settle out to align with neither vector exactly. So when we're thinking about the primary direction in which a system can be perturbed, we also have to recognize that not all species are going to respond the same to noise. So some species are going to be more sensitive to perturbations. For example, species with smaller population sizes at equilibrium are going to be more sensitive to demographic stochasticity. And some species might be more sensitive to environmental noise. So we can define a noise sensitive direction and think of how it relates to the dominant noise direction. So taking the same Jacobian we've been looking at, the linearization of our dynamics near some equilibrium, the dominant left eigenvector defines the direction from which perturbations decay the least efficiently or the most slowly. We call this the noise sensitive direction. And we can think of it as, map it out in this, again, 2D, just so I can draw it, space. So we have a CST direction, we have a noise sensitive direction. They're different unless the right and left eigenvectors happen to be equal. We have a primary noise direction that, again, I'll just draw in some arbitrarily different direction. And the relationship between the primary noise direction and the noise sensitive direction is also going to influence the strength of early warning signals that we can detect. So to show you this example, we'll give this angle a new name, theta. And to show you an example of this, in this example, we have a perturbation along a primary noise direction that is quite far from the noise sensitive direction. Whereas here we have perturbation in a primary noise direction that is much closer to the noise sensitive direction. And when the primary noise direction is closer to the noise sensitive direction, that perturbation is going to decay more slowly. Now, why do we care about that? Because remember, being perturbed away from the equilibrium and watching how it decays back is where we get our early warning signals. If we never leave equilibrium, we have no early warning signals. So the longer a system spends getting back to equilibrium following a perturbation, the more opportunity we have to observe early warning signals. So what we've seen so far is that early warning signals depend predictably on 4 vectors and 2 key angles between those vectors. So we have the CST direction, which is the dominant right eigenvector of the Jacobian that describes the linearized dynamics around some equilibrium point. We have the noise sensitive direction, that's the dominant left eigenvector of the same matrix. We have an observation direction, that is which species, or in higher dimensions, we could think of this as a plane, so maybe multiple species are being monitored. And the angle between the CST direction and the observation direction is the first one we examined, phi. And then we can have a primary noise direction and another angle that I'm calling theta between those. And the best opportunity to observe early warning signals is when both of these angles are small. So what makes both of the angles small? This was the question that he looked at. And he studied this problem not just in a 2-dimensional sort of toy model to build intuition, but in a formal sense using a model of arbitrary directions. So now X is a vector state variable. We have some, again, arbitrary function. We're going to linearize it. That's why I don't really care what this function is. We have some function describing the population dynamics of any number of interacting species, all subsumed in this vector state variable X. And we can approximate the dynamics near the equilibrium using an Ornstein-Uhlenbeck process, which I've written here. The part that's driven by the deterministic bit of the model is given by the Jacobian evaluated equilibrium. And the bit that's driven by the noise is given by a matrix B that describes the directionality in the noise, again, evaluated at equilibrium. And what Alex did is he used this OU process and calculated the steady state variance in our vector state variable X, how that depended on various key quantities. He looked at the distance from the tipping point, the angle between the CST direction and the observation direction, the other angle we were talking about, so the angle between the primary noise direction and the noise sensitive direction. But then he also looked at the strength of anisotropy in the noise. So how asymmetric is this matrix? How biased is it towards, in certain directions in the phase space? And then he also looked at how the variance in our state variable depended on the size of the system. In other words, the dimension of X, how many species we're modeling. And so we can summarize his results by thinking steady state variance in X along the blank direction is proportional to blank. So we're going to fill in the blanks a couple of different ways. We're first going to think about noise is initially, upon perturbation, going to grow in the primary noise direction and then decay over time, and it will decay most slowly along this noise sensitive direction. So we're sort of marching from a perturbation that went here. There's influence of the noise sensitive direction, and the steady state variance in our state variable X along the noise sensitive direction is proportional to the cosine of this angle between the primary noise direction and the noise sensitive direction. Then we can think about the influence of the critical slowing down direction, and the variance in this direction is proportional to the variance we already had over the square root of the magnitude of the dominant eigenvector. And finally, we can kind of project that down to our observation direction, which, if we're going to look for this in data, is the primary thing we're interested in. And we gain another factor here, which is the cosine of the angle between the critical slowing down direction and the observation direction. So this is pulling in all of those features that we sort of walked through one at a time in the two species example, now seeing how they build up to an I'm showing you in 2D again, just so I can draw it, but these are results that are general to any number of species, any number of dimensions in our state variable X. Alex was also able to derive that the steady state variance in our vector state variable X along the observation direction scales proportionately with the portion of the state variables being monitored. So if we have n species in the system, big N species in the system, and we're collecting data on little n of them, the larger little n is, the more variance we're going to be able to observe, and therefore the greater opportunity to observe a rise in variance and early warning signals. That's pretty intuitive. The more data you have, the more chance you have of seeing early warning signals, but we can show you how it scales. It scales linearly. And the steady state variance also scales with the proportion of the state space that's spanned by the strong noise sources. So sort of how much, if this were just straight horizontal, it would just be spanning sort of one species, and this M would be one. So how much of the state space is the noise allowing us to explore, is a way to think of this term. So these are the rules of thumb I presented already as a result of Amy's work. And now we can add to our understanding of when and where we can expect to see early warning signals of tipping points based on Alex's work. So now we can say even more and understand that factors that strengthen early warning signals in general, a small angle between the observation direction and the critical slowing down direction, meaning we should monitor the species whose axis is closest to the critical slowing down direction near the tipping point. We have a greater opportunity to see early warning signals if noise is isotropic. If it's anisotropic, so directional, then if there's a small angle between the dominant noise direction and the noise sensitive direction, again, that maximizes our opportunity to observe early warning signals. Likewise, if the dominant noise direction isn't that much stronger than the noise in the other directions, in other words, if we have anisotropic noise, but not too anisotropic, it's close to symmetrical, again, that maximizes our opportunity to observe early warning signals. It's easier to observe early warning signals in when the dynamics are of lower dimension, so fewer species in our system or strongly interacting species in our system. And this sort of concludes our theoretical understanding of this problem. What I'm going to do just briefly as I wrap up my talk is to show you how we've tested these insights on an actual nonlinear model. Everything I've shown you is based on linearization. So let's see how it works in a nonlinear model and what can we illustrate. So to do this, we looked at the Locke-Volterra competition model. The equation is shown down here. And we increased the competition coefficients, the alpha terms in the way I've written this model. As we increase those, we drive the system towards a tipping point. I've set up the parameter values so that species 1 experiences weaker intraspecific density dependence, and that is this K sub I is what we call the carrying capacity of, I'm sure this model is very familiar to some of you, but in case others haven't seen it, this is the carrying capacity for species I. And when that is larger, it means the environment can support more individuals. So smaller carrying capacity means more intense competition between individuals of the same species. So species 1 is experiencing weaker intraspecific and stronger interspecific competition. So the stronger interspecific competition comes in an asymmetry in how I set these alpha terms. Now I've selected these inequalities deliberately so that Amy's rules of thumb make it clear that we should monitor species 1 because it satisfies both rules of thumb. It's the either when we look at it, it's telling us we should monitor species 1. That should be our observation direction, and that will maximize our chance of seeing early warning signals. So what this model, we can look at the critical slowing down direction as we approach equilibrium. When we're far from the tipping point, then the two species nullclines intersect in a spot, and I'm going to put in a primary noise direction that increases the variance in species 2. So we have most of our variance, we're far from tipping. Critical slowing down is not really having an effect. Most of our variance is along this primary noise axis. As we get closer to the tipping point, the distribution spreads out and it gets closer to spreading out along the critical slowing down direction. And when we're very, very close to the tipping point, essentially all of our variance is collapsed across along the CSD direction here. So we can look at the variance in the population sizes as a function of our distance from the tipping point, which I'm showing you. I'm changing the competition coefficient alpha to drive the dynamics across the tipping or towards the tipping point, but I'm just sort of summarizing the effect of those changes by showing you the dominant eigenvalue approaching 0. And we know that the variance in the population sizes has to be bounded between the variance in the CSD direction itself, that's the most variance we can possibly observe, and the variance perpendicular to CSD direction. That's the worst observation axis we could choose. And if we simulate the model, of course, we did this multiple times. I'm just showing you one realization. These are the population densities in time as I'm gradually driving the system towards the tipping point. And we see an increase in variance in both species, but the increase in variance is stronger for species 2. I'm sorry, for species 1. And that is because species 1 has a smaller angle between its axis and the CSD direction than does species 2. And we see here that, in fact, species 1 gets us quite close to the maximum variance we could observe. So the maximum opportunity to observe early warning signals if we were to monitor species 1, just like the rules of thumb tell us. Now, there's one more interesting case I want to show you because it really drives home the purpose of all this. It's not just to understand it theoretically, but it really does inform how we should be monitoring for early warning signals in real systems. So let's look at the special case of equal competitors. So now I've set all of the parameter values equal for the two species. And so now, of course, I've labeled one species 1 and one species 2, but their increase in variance is identical, expected to be identical because they're identical species. And so again, this exactly in the CSD direction gives us the maximum variance we could observe. Perpendicular to the CST direction gives us the minimum variance we could observe. And this is the variance in, I'm just showing you species 1. Again, if I did species 2, it would only look different for just due to the stochasticity and the realization. The expectation would have to be the same because they're identical species. So I'm just showing you one species, arbitrarily labeled species 1 here. And again, it's not bad. It gets us pretty close to the CST direction. So this is what I showed you already, confirming it's still true. But here's where it gets interesting. In this special case, the CST direction is the difference, x1 minus x2. And perpendicular to that, of course, is x1 plus x2. And so the absolute worst thing we could do if we were monitoring for early warning signals would be to look at the total population size in this community, x1 plus x2, because there is absolutely no detectable increase in variance in this because it's completely perpendicular to the CST direction. So in a situation where we have identical competitors and in an applied setting, we might be sort of tempted to treat them as just all one population. They're very similar species. Let's just count the total. We would completely lose all ability to detect any early warning signals in that system. So understanding the CST direction is really crucial for designing any sort of monitoring scheme. So this is what I've already showed you. I'm going to add one more bullet point here, which is to tell you that what I've just shown is that these findings, which is all based on linearization, linear theory, are consistent with nonlinear simulations. I've just shown you a couple examples, but it seems to hold up in simulated nonlinear data. So we believe it would hold up in real systems as well. I would like to wrap up by thanking my funding sources, as well as collaborators, students, and faculty members that I talk about these ideas with a lot. I was going to show you serious pictures of everybody, but many of the people I wanted to acknowledge are shown here in this photo of a day several years ago when we carved pumpkins together for Halloween. This is the American tradition, create pumpkins like this for Halloween. And because we're approaching Halloween here, I wanted to show that to you as a silly slide, but a serious acknowledgement of all the fantastic students and colleagues who have helped me figure out these things. That's all I have for you, and I would be delighted to take any questions you have.
Thank you, Karen, for this nice talk. Questions, please.
Hi, can you hear me?
Yes.
Thank you, Karen. That was a really, really nice talk. Very well summarized of fairly complex dynamics of how CSD plays out. Thank you so much. So I had a couple of questions. One of them, I think you sort of answered towards the end. You know, if you did have multiple species data, you're suggesting one shouldn't just add them up and then, or, you know,
Yeah.
compute. But I, you know, obviously adding would be wrong here, but you know, is there some other combination of two species data that could be more sort of, you know, revealing of the underlying dynamics? Or do you recommend that one should first, of course, look at the CST direction and then, of course, take that component? So, but if we don't know the directions, how do we go about using multi-species data when we do have it?
Yeah. I feel like there's a number of questions just embedded there. That's a really great question. So if we know the CST direction, there is, you know, in this case, looking at the difference, which I don't think any field ecologist would think to do. But if you did that, that's where you'd see the strongest early warning signal. Of course, you would have no way to know that if you didn't know the underlying model. So really the reason that Amy did that study of so many Jacobians, just it's all linear, so it doesn't really matter what the underlying nonlinear dynamics are, but let's just look at all these different possible combinations of species effects on one another. And she was able to derive those rules of thumb shown here. For two species, we feel like, okay, let's say you're just monitoring, you have the resources to monitor one species, which one should you pick? This is not going to guarantee that you pick the right one, but it's going to certainly make it much better much better chance of detecting early warning signals than picking a species at random. For higher than 2 species, we don't have these types of rules of thumb, but I haven't added the analysis to this talk. I unfortunately don't have the figures and slides to show you. But another thing that Amy did is she took a 5 species food web from the literature that had been empirically parameterized. And she did this experiment of who should we monitor? What if we naively applied these rules of thumb that we made for 2 species systems to this new 5D system? Is it better than choosing a species at random to monitor? And it actually is much better. So that's just anecdotal, but we suspect these rules of thumb scale up. We just haven't proven it.
Thank you. Thank you so much.
Any more questions?
Hi, I just want to ask that when you measure the angle between the CSD direction and the observation direction, so it's just out of curiosity that if there is some relation between the difference between the angles that species 1 is making and species 2 is having with the CSD direction in the timing of tipping, can there be somehow any connection between the difference in these angles and the timing of tipping?
So because the 2 species are orthogonal, the 2 angles always have to add up to 90 degrees, right? So are you asking if it gets closer to one observation direction or the other with tipping?
I'm just asking if I observe the difference between phi and theta. Suppose the angle is phi for species 1 and theta for species 2. If I observe the angle, difference between these 2 angles, and can there be any connection with the timing of tipping? These difference can this difference have?
Yeah, I understand your question now. I don't know the answer. I don't think so, but I'm not positive that that's correct. We didn't look at that. Off the top of my head, I can't think of why they would be constrained to change in a certain way in approach to the tipping point, but I'm not positive. I should think about that more. That's a very interesting question. Thank you.
Anyone is having any other question? So Karen, I have a couple of questions. So,
Sure.
so these are multi-species model, right? And
Yes.
so with all the variables, you consider identical noise, or the noise can be a bit different, and one can also consider the cross correlation between those noises. So
yes. Yeah, so the noise was, let me go to my slide where we looked at, it's the next one, it's this one. Yeah, so I can't remember off the top of my head exactly how Alex measured this. He came up with a measure of anisotropy. It's too late for me to say this word, which is a measure of asymmetry in B. And so that's, I believe that the measure he used accounted for both dominance in any one direction as well as, well, no, I guess if they're tightly correlated, if the perturbations are tightly correlated between the species, then you would probably see greater symmetry because a big perturbation is big in multiple directions, right? So I think that's indirectly included here, but certainly digging more deeply into the, we looked more at what the mechanisms are that go into this Jacobian than we did into what goes into this B matrix, but we could just as well dig into this B matrix and think about it more biologically, which I think is sort of the basis of your question. We haven't yet done that, but that would be very interesting to do.
Okay, and then my second question is that, let's say you have a 2 species model, and here both these species that I understood, they're following similar time scale, right?
Yeah.
So there is no time scale separation. So let's say if one grows faster than the other, So if there is a time scale separation, how that will influence this result?
Yeah. Oh, that's a great question. Let me see if I can put up the two species. Yeah. So if there's a time scale separation, let's say there's a factor multiplying, it's a big number or a small number. Imagine a factor out front here. That would
We can't hear you actually. Can you see what is happening? We can't hear Karen.
Oh, you can't hear me?
Yeah, now we can hear you.
Okay. I'm sorry about that. So if we consider multiplying A and B by some factor that's either a small number or a big number, that would make species 1 dynamics slower or faster. Let's make species 1 dynamics slower. Let's say there's a time scale separation and species 1 is having slower dynamics, then A would be smaller than D. So we'd be in this lower triangle, and then B would be smaller than C. So then in that case, it would be, I guess, the slow species we want to monitor.
Okay. Yeah. We can't hear you again, actually. Now we can hear you again.
Okay. Did you hear any of that?
Yes. So the last point we missed.
Okay, so I said, kind of walking through the logic, it gets us down to where we'd want to monitor species 1, which is the slower species. But I think thinking about a timescale separation probably deserves a more careful answer than what I just gave it, but I hadn't thought about it, but thinking about it now off the cuff points us to monitoring the slower species.
Okay. And just the final question I have, I think so one of the major goals of this early warning signal analysis can be just to have some mitigation policy, because if we know that an indicator is giving us an indication that a tipping is ahead, so can we apply some mitigation strategy to prevent it or to delay it? So I just want to know your comment how this sort of analysis can be used in that direction.
Yeah, it doesn't directly improve our ability to mitigate. So you would still need to know what is the bifurcation parameter. So you could design a mitigation strategy that reverses its direction. The insights I've shown here today don't give us any new insights into what those mitigation strategies should be, but they maximize our chances of knowing that it's time to employ them.
Okay. Yes.
Can I ask one final question?
Yes.
It's sort of related to this. So what are your thoughts on, early warning signals as a predictive tool or being able to estimate the actual critical point? And something we have sort of failed so far, right? So in some sense, we got into early warning signals because we don't know where the bifurcation point is. But
Yeah.
can we ever go back to the previous question? Where is the bifurcation point? How can we sort of obtain some insights in a more quantitative way about that? Any general thoughts on that? Thank you.
That's the really frustrating thing about essentially all of this early warning signal stuff in my mind. We don't know where the bifurcation is, and we don't know how big of a signal, right? All of these early warning signals are directional, but there's no magnitude associated with them. So I'm talking about all the things that help us see a rise in variance. And I could do analogous, I could have gone through rise and autocorrelation. We can go through them one by one, but then none of this helps us know how big of a rise is for real. And yeah, I mean, anybody that has a better answer to that, I want to hear it, because I don't know. It is a real limitation. Yeah,
I agree. I mean, this has been sort of, the internal criticism of our own work that we go through. How do we do better than this, right? Yeah. Thank you so much. Yeah.
Yeah.
Thanks for also giving the talk at this really odd time for you and on a Sunday night.
No problem. I'm glad I could still do it. As I said, I apologize that I'm not there joining you in person, but it's been really nice to be able to participate. And I'm going to watch all the videos, but probably in the morning my time.
Thank you.
Thank you.
Thank you.
Anyone is having any other question? So if not, let us thank Karen for this wonderful talk. Thank you, Karen. Thank you very much.
All right. Thank you. Bye, everyone.